All these PowerPoints that I'm using for this course have been provided by the Waxman Student Scholars Program at Rutgers University. Today, we're going to be talking about Chapter 4, Sequencing DNA. In this case, we're going to spend some time looking at how sequencing is done and how it will apply to when you have to analyze a sequence. A little review from before. The transcription of the chromosomal DNA led to mRNA, which then Rutgers extracted from the plants and made our cDNA library. These were transformed into bacteria, and this is what became our library. We jump in this stage right here where we take colonies, we grow them, and we purify plasmids. When we purify them, we send them out for sequencing. Now, the plasmid is shown here, and if you remember from earlier videos, there's the SFIA and SFIB site where the insert was put. It was ligated in this section, and when it's ligated in this section, it is directional, which means that the SFIA region of the gene can only fit into the SFIA region of the backbone. That's important because we don't have to worry about having to reverse the sequences or having any other issues. When this is done, if you take a look here, this is an older slide where they have Wolffia DNA, but we're working with Landoltia punctata, and in this case you see that it's been ligated in this section. Here's our SFIA site, and if we look at the sequence, here we have the ATTA, CGG, CC, GGG, and then there are a bunch of ends. These ends indicate that when they ligate in the actual unknown, the gene of interest, we're not sure how many G's or how many guanines become added at this point. So when you get your sequences to analyze, I will tell you to start the sequence after the run of G's. This is what I'm talking about. Here is the forward primer. And the forward primer we use to sequence the DNA. Okay, just a little review. Determining the size of the insert by PCR and digest. Before we send our samples out to be sequenced, we need to verify with the PCR and the digest to see that this sequence is large enough to be sent out for sequencing, and if the sample has a bright enough band and enough DNA in the actual restriction digest. Remember that the PCR is done to the bacterial colony, and the restriction digest is done to the mini prep. The mini prep DNA is the tube, the yellow tube that's in the freezer that I'm going to send out, or I have sent out, to be sequenced by Rutgers University. So let's take a look at what's happening here. We analyze our PCR gel, and we see that the insert is roughly over 1,000 bases. It's between that area that's very difficult to predict how large the sample is. It could be 1,200, could be 1,250, we're not sure here. And if we look at our clone, our library clone, our uncut and cut DNA from the restriction digest, we notice that there are tons of bands. And if you remember, the vector is roughly 2.9 kilobases. So we expect that the vector, expect that when we cut the vector in restriction digest with PVU2, it will cut it on this side and this side typically. When we digest it, we will be left with two fragments. Those two fragments are shown here. Oops, sorry about that. Those two fragments are shown here. Here is the vector backbone at close to 3,000 bases, and the insert we see is actually, let me just take a look at that. It's actually right below here, and we notice that the band is actually showing up around 1650. So we know that that's the band. Now, if you remember, up here in the PCR, we were to subtract 200 bases for the overhang. So it's the insert plus 200 bases. Here, we have to subtract 700 because there are 700 kilobase or 700 bases that are not part of our actual unknown gene. These are part of the vector. So we don't want to take that into consideration when we're calculating our insert size. Okay, So what happens to that data? If it does correlate and the information is correct, then we should send it out for sequencing. Again, I'd have to look at whether the band is bright and there's enough DNA. If it's a very faint band, and typically, I'm going to go back a second, typically if the ladder is as bright, or if the sample is as bright as the ladder, then it's a good indication that that will sequence well. So we usually use that as a guide. Now, when was sequencing developed? First developed in the mid-1970s, and it rapidly scientists started rapidly sequencing genomes, working on things. Haemophilus influenza was found to have 1,700 genes, 1995. Later, yeast was sequenced. Then C. elegans was sequenced. And eventually, in 2003, we completed the human genome. And we found there that there were roughly 25,000 genes in the human genome. That was a shocker for most scientists, because we felt that humans must be so much more advanced, more developed than other organisms. We must have more genes. And what we found out is that no, our genes just do a lot more. We do have similar genes to other organisms that we've studied. So handling the explosion of sequence data. If you take a look, this is the amount of sequences in the database in GenBank, starting from the time that we started sequencing, and you could see there's a rapid explosion of the amount, and this number is much higher now. Okay, so what does that mean? It means that the database needed to be able to process and keep all that information. And GenBank is the National Institutes of Health Genetic Sequencing Database. It's a collection of annotated sequences, which means that you can't just deposit in there and have your information posted. It has to be checked and verified, and therefore the information that's in the database should not be junk or incorrect. So it keeps the integrity of the database. So if you take a look, genome sequencing project statistics, you see that in different types of organisms, there are all different numbers of projects. This is determined in 2009, but incredible from 2008 to 2009, the changes that occurred, and now the numbers are much higher. So, like I said, GenBank is part of NIH, and that project, the main important part of that is that GenBank makes the public have access to the DNA sequences. So if you discover something at a university or through this program, we're going to post it into NCBI and into GenBank, and that information is available for all scientists around the world to use. And it's a way for scientists to work together. So why are we sequencing these genomes? Basically, we'd like to be able to compare the organisms to other organisms in the database, to be able to see what organisms have similar genes, what their function and expression of the genes may be. There are many different projects that can come off of sequencing a genome. And again, we've been working on Lambda for the past couple of years, and that seems to be something that has been very, very good and very helpful. Once a gene is sequenced, a lot of the information can be determined about the gene, the number and location of all the restriction sites without restriction mapping. Okay, so now we have here, let's say, a sequence of a gene, and the only one of the things you need to understand is that when we get the sequence, we may not know what frame or reading frame we are in, and depending on the reading frame, you may get a different set of amino acids. So luckily, the programs that are designed to analyze this sequence on NCBI help us to determine if which frame is actually the best reading frame for this protein, or the most that has matches on the database. In 1980, Fred Sanger and Walter Gilbert developed methods for DNA sequencing for the 1970s experiments that they did. So they received the Nobel Prize in 1980. These methods are still being worked on today, but they're new. The older methods were a lot harder to handle and took a lot more time. Nowadays, our sequencing machines are much faster. So how does it work? The fundamental idea behind both methods is the same. You need a starting point. Okay, one end of the DNA is to be sequenced, And so you're only sequencing that one side of the template. And besides that, you need a primer to be able to begin sequencing. Remember that DNA polymerase needs something to hang off on in order to start adding nucleotides. So it needs a primer. And typically in your body or in organisms, RNA is the primer. In our purposes, it's going to be DNA. DNA fragments are generated at random lengths that end with either A, T, C, or G. And a random of population of DNA fragments are then separated using high-resolution gels or chromatography. The gel system can separate as fine as one base difference. So let's take a look. First clicker question, and again, these quizzes are in EDU 2.0. You can go there and take the quizzes. Sanger method makes use of DNA synthesis replication. DNA synthesis always starts by adding units to A. Just talked about it. Let's move on to more information. Now, no one really knows why we need to have a primer, but we do when we actually generate DNA. We know that the DNA polymerase needs to hang on to the primer. So what happens? The DNA is synthesized. Imagine that this is DNA polymerase building off of the template. DNA polymerase always synthesizes a new strand in a 5' to 3' direction. And here we have a nucleotide, and referring to the 5' end, here you have your 5' end where it can be bound to another nucleotide, or the 3' end where it can be bound. Okay, and we haven't really talked about this, but if I wanted to stop a sequence, let's say these elephants are all nucleotides, and I wanted to, and let's say I wanted to add nucleotides to this sequence. Okay, I add it to the 3' end, I add it to the 3' end. If I wanted to stop, make sure that the last elephant was always in A, what would I need to do? Let's say this elephant was in A, or this elephant is in A, and I don't want anything else to attach to it. What would I need to do? So basically, the nucleotides here, if we take a look at them, in this case is thymine, we're looking at the difference between deoxythymidine triphosphate, or the DTTP, or dideoxy. Okay, the dideoxythymine has one big difference. This one has a hydroxyl group on the 3' carbon, while the dideoxy has a hydrogen. So on this 3' carbon, I can add more nucleotides if I have a hydroxyl. But if I don't have a hydroxyl here, I can't add another base. So this is terminating the sequence. It will stop there. The last base, if this was in solution, would be a T, because it wouldn't be able to add anything else behind it. So again, here's my 3' hydroxyl group. There's a dehydration synthesis. Water is removed. The bond is formed. But if you don't have that hydroxyl group, you cannot do this. So what would happen if we carry out synthesis and we add DDC, dideoxy Cs, instead of the regular cytosine? So the dideoxy, if you remember, is back here. Dideoxy has no hydroxyl group. Deoxy has a hydroxyl group. Go to your quiz and try to answer the question. You may want to pause the video as you answer the question so you can take a minute to think about it. Here's another question. Now, instead of just adding DDCs, we add DDG instead of DG. What would happen? And what happens if you add equal amounts of these? So in the other two questions, we weren't adding any of the original base.